students. Now I am talking about the Kreb cycle. The Kreb cycle has a total of 8 metabolic reactions involved in the full oxidation of our food molecules into carbon dioxides. Carbon dioxides CO2. Okay, this is cycle of citric acid cycles. Its another name is TCA cycle, tricarboxylic acid cycle. First step, pyruvate. Pyruvate is converted into acetyl-CoA. Acetyl-CoA is a removal of COA. Okay. Next one, acetyl-CoA is converted into citrate. Citrate converted into cis-agoninate. Cis-agoninate converted into diisocitrate is including water H2O. Diisocitrate converted into alpha-ketoglutarate. Next, alpha-ketoglutarate converted into succinyl-CoA. Succinyl-CoA is converted into succinate. Succinate is converted into fumarate. Fumarate converted into malate. Malate converted into oxaloacetate is repeated in this citrate cycles. Okay. Next, enzymes are involved. First enzymes agonidase. Next agonidase, same enzymes. Third one isocitrate dehydrogenase. Fourth one alpha ketoglutarate dehydrogenase. Fifth, succinyl-CoA synthase. Succinyl-CoA is converted into succinate is help of the enzymes succinyl-CoA synthase. Next, succinate is converted into fumarate with the help of enzyme succinic dehydrogenase. Next, fumarate is converted into malate with enzymes fumarase. It include water. Next one, malate is converted into oxaloacetate with the help of the enzymes malate dehydrogenase. NAD plus converted into NADH plus H plus. Next, Kreb cycle reactions. 8 major reactions occur in the Kreb cycles. First one, it's a metabolic cycle because it regenerates the same metabolite at the end that start with oxaloacetate. It's a chemical structure of oxaloacetate. Next, the Kreb cycles shown enzymatically is a help of the enzymes which involved in the Kreb cycles. First one, citrate synthase agonidase isodihydrogenase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase. Next one, succinyl-CoA synthase, succinate dehydrogenase, fumarase, malate dehydrogenase. Okay. Stepwise involved in the TCA cycle. First one, the 8 reactions, as already I said told, 8 reactions in the metabolic reactions of Kreb cycles. First one, the 8 reactions of the citric acid cycles is a small molecules. This is oxaloacetate. This is a small molecules oxaloacetate. This is a starting place in the reaction of the cycles. The cycle starts by addition of an acetyl group to oxaloacetate. Then in 8 steps, the acetyl group is completely broken about restoring the oxaloacetate molecules in the another round. First round is the starting place oxaloacetate. Next, restoring this molecules oxaloacetate. In a typically biological tweets, it's not quite this simple. You might imagine that the enzymes could just pop off 2 carbon atoms of the acetyl group using the oxaloacetate. Next, however, be carefully labeling particular carbon atoms in these molecules. Scientists have found that things got shuffled around a bit. 2 carbon atoms in the original oxaloacetate are the parts of the actually released as carbon dioxides. Then at the end of the cycle, the original acetate atoms shuffled around recreate the oxaloacetate. The first one, starting place is oxaloacetate. Next, end place is also known as oxaloacetate. Reaction 1, citrate synthase, oxaloacetate plus acetyl-CoA is converted into citrate. So help of the enzyme citrate synthase. Next reaction 2, agonidase. Citrate is converted into isocitrate with help of the enzymes agonidase. Okay. Third one, isocitrate dehydrogenase is a chemical structure of isocitrate converted into alpha ketoglutarate. Help of the enzymes isocitrate dehydrogenase. CO2 is released and the molecule NADH formed in this process. Next, isocitrate dehydrogenase. The reactions mechanisms involve NAD plus and Mg2 plus, magnesium 2 plus as a cofactors. Next, isocitrate dehydrogenase uses NAD plus Mg2 plus and the two molecules are cofactors within the reactions mechanisms that is shown in the next few slides. Isocitrate dehydrogenase. The step 1 details the dehydrogenase activity of the enzymes. First, an acidic residues in the active site deprotonates the alcohol positions of isocitrate. This is a carbonyl functional group. This enables the extractions of the electrons and the hydrogens from the C2 carbon positions by the NAD plus cofactor. The proton is released into the environment by the acidic residue in the active site when NADH excites the active sites. NAD plus is converted into NADH plus H plus. Next, decarboxylase and lyase activity. In this step 2, decarboxylation occurs. Is cofactors Mg2 plus cofactors are stabilized. Next, carboxylic acids is the functional group. Next, ketone formations. This one is ketone formations. As is the carbonyl electrons. This is a ketone functional group. This is a pyruvate dehydrogenase complex and that of the glyceraldehyde 3-phosphate is a glycolysis cycles. Which enzymes are used? Dehydrogenase enzymes are used. This is the glycolytic pathway. This is for many different ways. Dehydrogenase enzymes can be worked to oxidize into the biological molecules. Next, react 4. Alpha ketoglutarate dehydrogenase. Alpha ketoglutarate is converted into succinyl-CoA. Okay, NAD plus has converted into NADH plus H plus. This is alpha ketoglutarate dehydrogenase. These are pyruvate dehydrogenase enzymes that alpha ketoglutarate dehydrogenase also uses. COSH substrate TPP, lipoamide, FAD and NAD plus is a cofactors are used in the TCA cycles. Okay, this is the E1 complex and E2 complex and E3 complex. This E1, E2, E3 are subunit in the Kreb cycles. Okay. Fifth reaction, succinyl-CoA is converted into succinate with the help of the enzyme succinyl-CoA synthase. This is substrate level phosphorylation. Reaction 6, succinate dehydrogenase. Succinate is converted into fumarate with the help of the enzyme succinate dehydrogenase. FAD converted into FADH2. Reaction 7, fumarate is converted into malate. Okay, this enzyme is also hydrolyzed as water is incorporated into the final structure. Reaction 8, malate dehydrogenase. L-malate is converted into oxaloacetate with the help of the enzymes malate dehydrogenase. One final energy carrier NADH and H plus is formed. NADH plus H plus is formed in these reactions. Reactions 8, malate dehydrogenase. Energy yield in the Kreb cycles 3 NADH, 1 FADH2, 1 GTP. This is the ATP equivalence. Next one the 2 per glucose, 2 cycles and 2 acetyl-CoA. 6 NADH plus, 2 FADH2, 2 GTP. Overall, the total energy yielded in one turn of the citric acid cycles are 3 molecules of NADH and H plus, 1 FADH2, 1 GTP. This is double for the energy potential in one glucose molecules as 2 molecules of acetyl-CoA with enter the Krebs cycle. The major regulatory steps: pyruvate dehydrogenase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase. Okay, regulations of this pathway is mediated primarily by 3 of the major dehydrogenase enzymes in the pathway. These include pyruvate dehydrogenase, which serves as the gatekeeper period to the beginning of the Krebs cycle, followed by the next 2 dehydrogenase and carboxylase steps in the pathway, mediated by isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. Essentially, all of these steps where CO2 is released. This is carbon dioxides are released. All of these enzymes are highly regulated by the energy load of the cell. Next, pyruvate dehydrogenase complex. This is NADH inactivation, acetyl-CoA inactivation, ATP, adenosine triphosphate is inactivations, ADP activation. This is the allosteric regulations. Next, phosphorylations. Next, isocitrate dehydrogenase. Low energy activities this pathway, high ADP levels, high energy, high ATP, high NADH. This is alpha ketoglutarate dehydrogenase. This is high ATP, high NADH. Following to the alpha ketoglutarate enzymes undergoes the negative feedback incubations by accumulation of these succinyl-CoA product levels. High levels of ATP and NADH are also inhibitory. Notes: this vitamins deficiency can cause diseases states such as beriberi. A lack of vitamin B1 will lead to inefficient production of TPP cofactor required for BDH and KGTH complexes. Lack of vitamin B1, inefficient TPP, reduced capacity of PDH, as a complex of alpha ketoglutarate and DH. This already I said told alpha ketoglutarate dehydrogenase which enzymes are used. Okay, next one, thus these enzymatic systems do not function well the reproduce of ATP. So patients will experience weight loss, emotional distributions and other neurological and muscular symptoms. So weakness and pain in the limbs, periods of irregular heart rate. Thank you.